about is genetic screening. Genetic screening is the application of a test on people for the systematic early detection or to determine whether a person carries a predisposition which may produce a hereditary disease in their offspring. So guys, we have two types of genetic screening. First is the carrier screening. It's a genetic test used to determine if a healthy person is a carrier of recessive genetic disorder. For the keyword is it involves family, individual, and potential parents. For the second one it is a prenatal screening or prenatal screening. Takes for chromosomes abnormal. For further explanations about the two types of genetic screening, let's hear it from Dr. J.
Here are some examples of prenatal screening. First is Down syndrome. Down syndrome is a congenital disorder arising from a chromosome defect, causing intellectual impairment and physical abnormalities including short stature and a broad facial profile. It arises from a defect involving chromosome 21, usually an extra copy or the trisomy 21. Next is hemophilia, a medical condition in which the ability of the blood to clot is severely reduced, causing the sufferer to bleed severely from even a slight injury. The condition is typically caused by hereditary lack of coagulation factor, most often factor VIII. The third is the spinal bifida, a congenital defect of the spine in which part of the spinal cord and its meninges are exposed through a gap in the backbone. It often causes paralysis of the lower limbs and sometimes mental handicap.
Good morning, I'm Dr. Y from Sidom Institute and I'm going to discuss about transcriptomics. Transcriptomics. When we say transcriptomics, it is the study of transcriptome. And what is transcriptome? Transcriptome is the complete set of RNA transcripts such as mRNA, tRNA, but it varies from one cell to another cell. Unlike genome is the complete set of DNA from an organism, but it does not vary from one cell to another cell. If you wonder why liver cells, nerve cells, muscle cells, blood cells are different from each other, it is because of transcriptome.
The process of transcriptomics. A transcriptome captures a snapshot in time of the total transcripts present in a cell. Transcriptomics technologies provide a broad account of which cellular processes are active and which are dormant. A major challenge in molecular biology lies in the understanding how the same genome can give rise to different cell types and how gene expression is regulated. Measuring the expression of an organism's genes in different tissues or conditions, or at different times, gives information on how genes are regulated and reveal details of an organism's biology.
Good morning everyone, I am Dr. D to discuss to you what is microarrays. Microarrays are grids of DNA segments of known resource that is used to test and map DNA fragments, antibodies, or proteins. Microarrays are used to determine what gene is expressed by a cell or the tissue. To explain more about microarrays process, let's hear it from Dr.
The process of microarrays. Microarrays are used to determine an expression of a tissue or a cell. DNA of a cell or tissue will be isolated and extracted to get the complementary DNA, or also called cDNA. Microarrays have thousands of known sequences of DNA. Scientists acknowledge a complementary DNA based on their assigned color. Commonly, complementary DNA will stick to its complementing strand in the microarray. Red are commonly the complementary DNA that has a problem, while the normal complementary DNA, green, is its color.
Hi, I'm Dr. C and I'm going to discuss about genome sequencing.
So first, let's define what is a genome. So a genome is the complete set of DNA present in an organism, including all of its genes. This means that it contains all of the information needed to build and maintain an organism. So when we say genome sequencing, it is the process of figuring out the order of nucleotides within a DNA molecule, or bases in the genome. These nucleotide bases are adenosine, cytosine, guanine, and thymine. For example, is in human genome, it is made up of 3 billion of the genetic letters. It is often referred to decoding, but a sequence is still very much in code, in the sense that a genome is simply a very long string of letters in a mysterious language.
Imagine a passage in a textbook without punctuations. No breaks between words and with strings of scattered letters between these words or sentences. It is hard for us to understand or pick out the meaning of the passage unless we highlight these important words. We can compare this in sequencing a genome, wherein sequencing the genome does not lay open the genetic secrets of an entire organism. There's much work remains to be done that scientists still have to translate those strings of letters in an understanding of how the genome works. So sequencing a large or an entire genome is hard, so it is mostly done by high-tech machines.
So here's Dr. J to discuss on how to sequence a genome.
How to sequence a genome. There are different ways or methods of DNA sequencing, but the actual process of DNA sequencing is the same. Sequencing employs a technique known as electrophoresis to separate pieces of DNA that differ in length by only one base, clone by clone, where the genome will be cut first into large chunks called clones. Then genome mapping techniques will be used, then cut it again into smaller clones, and finally, the pieces will be sequenced and use the overlaps to reconstruct the sequence of the whole clone. Whole genome shotgun, where the genome will be broken into small pieces, sequencing the pieces and reassembling the pieces into the full genome sequence. Each of these approaches has advantages and disadvantages. The clone-by-clone method is reliable but slow, and the mapping step can be especially time-consuming. By contrast, the whole genome shotgun method is potentially very fast, but it can be extremely difficult to put together so many tiny pieces of sequence at all at once.